Welcome to this scientific lecture on high-throughput experiments in drug discovery using droplet microfluidics. Over the next 10 minutes, we'll explore how miniaturized droplet systems are revolutionizing pharmaceutical research. Let's start by understanding the fundamental challenge in drug discovery. Traditional pharmaceutical research is extraordinarily slow and expensive. On average, developing a new drug takes 10 to 15 years from initial discovery to market approval. The financial burden is staggering, exceeding $2 billion per successful drug that reaches patients. Perhaps most discouraging, the success rate is less than 10%. 9 out of 10 drug candidates fail during development. You might ask, why is drug discovery so challenging? The answer lies in the sheer complexity of biological systems and the need to test millions of potential drug candidates. Traditional screening uses large test tubes or well plates. Each experiment requires milliliters of precious reagents, limiting throughput and driving up costs dramatically. This is where droplet microfluidics enters as a transformative technology. It's a method that manipulates fluids at the microscale, creating tiny droplets that serve as isolated reaction chambers. What exactly is droplet microfluidics? Think of it as plumbing for liquids at the microscopic scale. Imagine channels etched into glass or polymer chips, with widths ranging from 10 to 100 micrometers. That's thinner than a human hair. Within these channels, we generate discrete droplets. Each droplet ranges from 1 to 100 picoliters. A picoliter is 1/trillionth of a liter. To put this in perspective, a single droplet is about 1 million times smaller than a typical water droplet. How are these microscopic droplets actually generated? The answer involves precise control of immiscible fluids. We use two immiscible phases, typically an aqueous solution containing our biological samples and an oil phase with surfactants. At a junction, the aqueous phase is pinched off by the surrounding oil, forming monodispersed droplets. Surfactants are crucial. They're molecules that stabilize the droplet interface, preventing coalescence and ensuring each droplet remains an isolated microreactor. Now you might wonder, why go through all this trouble to create tiny droplets? What advantages do they offer for drug discovery? First, miniaturization dramatically reduces reagent consumption. Instead of milliliters, we use picoliters. This means we can perform millions of experiments with the same amount of material that would yield only hundreds in traditional formats. Second, throughput increases exponentially. Droplet systems can generate and analyze thousands of droplets per second. This translates to screening millions of conditions per day. Third, each droplet acts as an isolated microenvironment. There's no cross-contamination between experiments, and we achieve precise control over reaction conditions. Let's quantify this. The volume ratio between 1 milliliter and 1 picoliter is 10 to the 9th power. That's a billion-fold reduction in reagent volume. Let me give you an analogy to appreciate this scale. Imagine traditional screening as testing vaccines by injecting entire swimming pools into patients. Droplet microfluidics is like testing with single drops instead. Same information, billion times less material, and you can test billions of variations simultaneously. Let's dive deeper into the technology. How exactly do we generate these droplets with such precision? The most common approach uses a T-junction geometry. The aqueous phase flows through one channel, meeting the oil phase flowing perpendicular. At this junction, interfacial forces cause the aqueous stream to break into uniform droplets. The result is a train of monodispersed droplets, each identical in size and composition. This uniformity is critical for reproducible experiments. An alternative is flow-focusing geometry, where the aqueous phase is squeezed from multiple directions by converging oil streams. This provides even better control over droplet size. Now let's explore specific applications in drug discovery. How are these droplet systems actually used to screen drug candidates? First, enzyme inhibitor screening. Many drugs work by inhibiting specific enzymes involved in disease pathways. Droplet systems allow rapid screening of millions of compounds against target enzymes. Each droplet contains enzyme, substrate, and a potential inhibitor. Fluorescent readouts report enzyme activity. Droplets showing reduced fluorescence indicate successful inhibition. Second, cell-based assays. We can encapsulate single cells in droplets along with drug candidates and monitor cellular responses. This is revolutionary because it provides single-cell resolution. We can observe how individual cells respond to drugs, capturing heterogeneity that bulk assays miss. Third, protein crystallization for structural biology. Determining drug target structures requires protein crystals, which are notoriously difficult to grow. Droplet microfluidics enables screening thousands of crystallization conditions with minimal protein. Each droplet is a separate crystallization trial, dramatically accelerating structure determination. Let me share a compelling real-world example. Antibody therapeutics represent one of the fastest-growing drug classes. Discovering effective antibodies traditionally requires months of work. Traditional methods screen thousands of antibody variants per week, using robotic systems and multi-well plates. Droplet microfluidic systems can screen millions of variants per day. This thousand-fold increase in throughput compresses months of work into days. This acceleration is transformative. Researchers can explore vastly larger chemical and biological space, increasing the probability of finding optimal drug candidates. A critical question is, how do we actually analyze what's happening inside millions of individual droplets? Fluorescence-based detection is the workhorse method. Droplets flow past a laser interrogation point where fluorescent signals are measured at kilohertz frequencies. As each droplet passes through the laser beam, emitted fluorescence is captured by detectors. This generates a real-time readout of each droplet's contents and activity. High-speed cameras complement fluorescence detection, providing visual confirmation of droplet behavior, uniformity, and any anomalies. Perhaps most powerful is the ability to sort droplets based on their fluorescent signals. Droplets of interest can be deflected into collection channels using electric fields or acoustic forces. This creates a closed-loop system. Generate droplets, screen them, identify hits, and physically recover those hits for further validation. It's like having millions of tiny test tubes that you can individually inspect and sort. Let's quantify the performance advantages with concrete numbers that highlight why this technology is so impactful. Modern droplet systems achieve throughput of 10,000 droplets per second. Operating continuously, that's over 800 million droplets per day. At 10 picoliters per droplet, 800 million droplets consume only 8 milliliters of total reagent. In traditional 96-well plates, this same volume would yield fewer than 100 experiments. The cost reduction exceeds 1,000-fold. For expensive biological reagents like antibodies or purified proteins, this makes previously impossible experiments economically feasible. Now, in the spirit of scientific rigor, we must address the challenges and limitations. No technology is perfect, and droplet microfluidics has its constraints. First, surface effects dominate at the microscale. Surface-to-volume ratios are extremely high, meaning molecules can adsorb to interfaces, potentially affecting reaction kinetics. Second, incubation times are often limited to minutes or hours. Long-term cell culture experiments spanning days become technically challenging. Third, the technology requires specialized equipment and expertise. Chip fabrication, fluid handling, and data analysis demand interdisciplinary knowledge spanning engineering, chemistry, and biology. Fourth, hits identified in picoliter droplets must be validated in larger volumes. The transition from droplet to bulk can introduce artifacts. Looking forward, where is this field heading? What innovations are on the horizon? First, integration with artificial intelligence and machine learning. AI algorithms can analyze the massive datasets generated, identifying subtle patterns and predicting optimal drug candidates. Second, multi-step reactions in droplet networks. Researchers are developing systems where droplets can be merged, split, and routed through complex networks, enabling sophisticated chemical synthesis and biological workflows. Imagine droplets as programmable microreactors, automatically executing complex experimental protocols without human intervention. Third, integration with organ-on-chip technologies. Combining droplet generation with three-dimensional tissue models could revolutionize toxicity testing and personalized medicine. Fourth, miniaturization toward point-of-care diagnostics. Portable droplet devices could enable rapid disease diagnosis in resource-limited settings, detecting rare biomarkers with extraordinary sensitivity. Let me share a timely case study. During the COVID-19 pandemic, droplet microfluidics played a crucial role in rapid drug repurposing efforts. Researchers used droplet systems to screen thousands of FDA-approved drugs against the SARS-CoV-2 virus in a matter of weeks, a process that would have taken months using conventional methods. Several promising candidates were identified and advanced to clinical trials. The speed advantage was literally life-saving, potentially accelerating therapeutic options during a global health crisis. This exemplifies the power of high-throughput droplet screening, compressing timelines when every day matters. For researchers considering implementing droplet microfluidics, what practical considerations should guide them? First, start with well-defined biological questions. The technology is a tool, not an end in it. Ensure your scientific problem genuinely benefits from high-throughput miniaturization. Second, invest time in robust assay development. Fluorescent reporters must be sensitive, specific, and stable within the droplet environment. Pilot experiments are essential. Third, weigh commercial platforms versus custom-built systems. Commercial options offer ease of use but less flexibility. Custom systems provide ultimate control but demand significant engineering effort. Fourth, assemble interdisciplinary teams. Success requires expertise in microfluidics, chemistry, biology, and data science. Collaboration is key. Let's summarize the key concepts we've covered in this lecture. Droplet microfluidics miniaturizes experiments to the picoliter scale, enabling billion-fold reductions in reagent consumption. Throughput increases dramatically, with systems capable of millions of experiments per day compared to hundreds with traditional methods. Applications are diverse, spanning enzyme inhibitor screening, cell-based assays, protein crystallization, and antibody discovery. Integrated detection, analysis, and sorting create closed-loop workflows that identify and physically recover hits. The technology demonstrably accelerates drug discovery timelines, compressing months into weeks or days. Challenges remain, including surface effects, limited incubation times, and the need for specialized expertise. Future innovations incorporating AI, complex droplet networks, and organ-on-chip integration promise even greater capabilities. Finally, let's consider the bigger picture. Why does this technology matter beyond the technical details? Droplet microfluidics democratizes access to high-throughput screening. Academic labs with limited budgets can now perform experiments previously restricted to pharmaceutical companies with million-dollar robotic systems. It enables exploration of vastly larger chemical and biological space, increasing the probability of discovering breakthrough therapeutics. Most importantly, it reduces the time from initial discovery to patient benefit. In medicine, speed matters. Faster drug discovery means earlier treatment options for patients facing life-threatening diseases. We're not just making experiments faster and cheaper. We're fundamentally changing what's possible in pharmaceutical research, opening doors to discoveries that were previously beyond reach. As we conclude this lecture, I want to leave you with a final thought. Droplet microfluidics represents a beautiful convergence of physics, chemistry, engineering, and biology. It reminds us that transformative progress in biomedicine often comes from applying principles from one field to solve problems in another. Technology alone does not drive progress. Rather, creative application of fundamental principles, combined with deep understanding of biological problems, yields breakthroughs. The future of drug discovery is undoubtedly interdisciplinary, and droplet microfluidics exemplifies this paradigm. Thank you for your attention. I hope this lecture has given you a comprehensive understanding of how these tiny droplets are making a big impact on human health.